A, are you there? A, are you there?
Yes.
Good. Now we stopped here at the stage of the hybridoma cell. This hybridoma cell, before it is put in a culture and allowed to mass produce, first it is screened in order to make sure that the specific antibody is being produced. Second, it goes through a process known as humanization. What is humanization? It is some sort of genetic modification. We will genetically modify the genes of the hybridoma cell to make it more human, so that it does not induce an allergic reaction when it is injected in your body. So this was the answer to your question. You said, when these hybridoma cells are injected into your body, how does your body not respond? Because before they are injected, they are genetically modified, and you don't have to know the details of this genetic modification. They are genetically modified and they are humanized. Easy?
Yes.
Good. So again, the making of the monoclonal antibodies, we make them in a very easy way. First, we will get a mouse. This is my mouse. I will inject this mouse with my what? Pay attention with me. The antigen. The mouse immune system will become activated, and then the mouse will produce what?
The plasma cells, and then we fuse them with the tumor cells.
Very good. These plasma cells are fused with tumor cells to make hybridoma cells. These hybridoma cells, before they are cultured in order to mass produce, they will first be screened to make sure that they are producing the requested or the desired antibody, and they are genetically modified, they are humanized, so they don't induce an allergic reaction. And after we screen them and we do the genetic modification we need, we will put them in a suitable culture, and we will allow them to multiply and multiply and multiply. Every time they multiply, they will produce lots of antibodies, and these antibodies will be used in medicine, and they will be used to make monoclonal antibodies. Easy?
Yes.
Good. Now, monoclonal antibodies, their uses. They are used in passive immunity. Clear?
Yes.
Good. They are used in the diagnosis and the prognosis and the treatment of disease. Now, how is that? Look, this is my monoclonal antibody that I made in the lab. This monoclonal antibody, I can attach it to a fluorescent marker. Fluorescent marker. What is a fluorescent marker? A fluorescent marker is a molecule that releases small amounts of radiation. So this molecule now is known as a biological fluorescent marker. Again, I will make the monoclonal antibody. And when I make it, this monoclonal antibody, I can attach it to a fluorescent marker. I am drawing something like a flag, okay? This fluorescent marker, from the name, it's a biological marker that produces small amounts of radiation. Okay, Aya?
Okay.
This monoclonal antibody, I will put it inside the body of the patient. When it goes inside the body of the patient, it will attach to a specific antigen. Yes?
Yes.
Good. So now don't forget, it is attached to the antigen, and it is also attached to the fluorescent marker. This patient is usually exposed to some sort of camera. He will enter inside a machine and he will be exposed to a gamma radiation camera. This camera releases light. When the light falls on the monoclonal antibody with the fluorescent marker will shine, and we will know the location of the antigen inside the patient's body and how much of the antigen is present. This helps us in the diagnosis of lots of diseases. Again, I want to know if a patient has cancer, God forbid. I will take monoclonal antibody. This is an antibody against the cancer antigen. This monoclonal antibody will be attached to a fluorescent marker, to a marker. Okay? fluorescent marker from the name. It's a small molecule that is attached to the monoclonal antibody that releases radiation, that releases fluorescence. Okay, Aya?
Okay.
I will put it inside the patient's body. This patient that has cancer. The monoclonal antibody will go bind with the cancer antigen. This is the monoclonal antibody that has the fluorescent marker, and it has the antibody receptor that will bind with the antigen. This is the cancer antigen. This fluorescent marker will then shine when this patient is exposed to some sort of camera, and the camera releases light. When this light falls on the fluorescent label on the biological or the monoclonal antibody, it will shine. It will mark it. And this will appear on a screen. And you will know from this test, this screening test, where is the antibody found in the patient's body and how much of it is present. So you can know if this patient has cancer and the stage of the cancer. Okay. Of course,
Okay.
too advanced, but you are studying just introduction to how things happen. This is the cancer cell here, and these are the monoclonal antibodies. These monoclonal antibodies, they are attached to another molecule known as the fluorescent marker. To label it, we want to just find it. It will be put inside the patient's body, and when they are inside the body, they will bind to a specific antigen, which is the cancer cell. And by using certain machines, we will know where is the monoclonal antibody. When we know where is it, we will know where is the cancer in the body and where did it spread into. Okay? Easy? Understood?
Yes.
Good. Now, because the monoclonal antibody can attach to other molecules, we can attach the monoclonal antibody not to a fluorescent marker, but to the medicine itself. I will attach the monoclonal antibody to the medicine. This monoclonal antibody will enter the body of the patient. And when it enters the body, it will bind with the antigen. So it has the medicine attached to it, and it will, it will bind, it will bind with the antigen. And this medicine will kill the antigen, will kill the pathogen. So this is another use of monoclonal antibody. It can be used for localized or customized treatment. This is a very, very advanced form of treatment because this type of treatment here will take the medicine to the sick cells, and the other cells of the body will not be affected by the medicine. And this will spare the side effects of the medicine. Normally, when you take a medicine and you put it inside your mouth, it affects the whole body. It will affect the healthy cells and the sick cells. That's why any medicine has side effects. But when the medicine goes only to the sick cell with the antigen, it will spare all the healthy body cells and you will have no side effects. Very localized treatment. And this is also used in cancer treatment. See, it's too advanced. Understood?
Yes.
Good. So I just want you to understand how the monoclonal antibodies are produced. So do you understand this diagram here? We inject the mouse, the immune system of the mouse will become activated. We will produce plasma cells. We will fuse them with tumor cells so that they can multiply and give you hybridoma cells. We will screen it first and we do some genetic modification first, and then they will produce on a mass lots of monoclonal antibodies. Is this clear?
Yes.
The monoclonal antibodies can be used in passive immunity. Clear?
Yes, miss.
Good. The monoclonal antibody can be attached to a fluorescent marker, and this will help in the diagnosis and the prognosis of disease. Where is the antigen in the body? How much of it is present in the body? So this will help us in the diagnosis and the prognosis of disease. Clear?
Yes.
Good. When this monoclonal antibody is attached to a medicine, then it will help us in the treatment of diseases. Clear?
Yes, miss.
Good. We understand how it works. Now, specific examples in your syllabus, the monoclonal antibody can be used to find out where is a blood clot formed when a patient has deep venous thrombosis. Deep venous thrombosis is a disease in which a blood clot happens in the deep veins of the legs, as you can see here. So what happens is that we will get our mouse and we will inject this mouse with our antigen, which is the fibrin protein. So the mouse will produce plasma cells or antibodies against the fibrin, mono antibodies against the fibrin, anti-fibrin antibodies, anti-fibrin antibodies. Are you with me?
Yes.
Good. The monoclonal antibody will be attached to a fluorescent marker. We will put it inside the patient's blood and they will go attach to the fibrin in the blood clot. And this will help us locate where is the blood clot and how big it is so we can treat it. Okay? Monoclonal antibody also helps us locate cancer cells and clots in the body. This is very important. Some people have clots in the body and we don't know which part of the body. So we inject them with monoclonal antibodies that are anti-fibrin. They will go bind to the fibrin and we will immediately know where is the clot. The advantages of using monoclonal antibodies, they're very fast, they're very quick, they're very accurate. Okay, Aya?
Okay.
Also, monoclonal antibodies can help us to diagnose HIV. What happens? And it helps us also with pregnancy test. Now, this is a test plate here that contains the HIV antigen. This is a plate that contains the antigen, the HIV antigen. Don't get confused. This is the HIV antigen. On this test plate, we first add drops of the patient's blood. This blood, if he is HIV positive, this blood will contain HIV antibodies. And then we wash it to remove the excess blood. And after we add a drop of blood, we add monoclonal antibodies. These monoclonal antibodies, they go and bind to the HIV antibodies. So the monoclonal antibodies, their antigen is the HIV antibodies if present. Okay, Aya? So this is the test plate. This test plate contains HIV antigen. We will add a drop of blood. If this patient is HIV positive, the HIV antibodies will go bind to the antigen. And then we will add monoclonal antibodies on top. These monoclonal antibodies, they bind with the HIV antigen if it is present. So it will help us mark and know if this patient is HIV positive or not. Okay?
Okay.
This monoclonal antibody is attached to an enzyme. This enzyme, it catalyzes a reaction that will result in a color change. So if this color change happens, we know that this patient is HIV positive. So the idea and why monoclonal antibodies are very helpful is that the monoclonal antibody can bind to a lot of things. We can bind it to a marker. We can bind it to an enzyme. We can bind it to a medicine. And it will go direct. We know that this monoclonal antibody will go attached to the antigen. Either we will mark the antigen, or we will treat the antigen and kill it, or we will send an enzyme to the antigen to do some type of reaction. Clear?
Clear.
Good. This is the idea. Here is the test plate. These are the HIV antigens. And then we add a drop of blood. If the blood contains HIV antibodies, they will go and attach themselves to the antigens. And then we wash this test plate to remove the excess blood. And then we attach, and then we add monoclonal antibodies that are attached to an enzyme that will go attached to the HIV antigen. So they are all attached to each other. And this would result in a color change. And if this color change happens, then this is an indication that this patient is HIV positive. And this is also used in pregnancy test. If you've ever seen a pregnancy test, it looks like this. It's a thin plastic strand with a window. Okay? And then you put a drop of urine here. This is urine. In the filter paper inside the pregnancy test, there are the monoclonal antibodies. They are already there. If this urine contains pregnancy hormones, So the urine will be absorbed up the filter paper. This is filter paper. If the pregnancy hormones are present, the monoclonal antibody will bind to the pregnancy hormones. And if they bind to the pregnancy hormones, there will be a color change. So the pregnancy test will show you 2 strips. 2 strips means positive. 1 strip means the pregnancy hormones are not present and this female is most probably not pregnant. Okay, Aya?
Okay.
Very good. So the whole idea of the pregnancy of the monoclonal antibodies, how they're done, I think it's easy. Understanding their function is understanding that the monoclonal antibody can bind to anything. They can bind to a fluorescent marker, so we will mark the antigen. They can bind to a medicine, they can bind to an enzyme, we can make it bind to anything we want. This is the whole idea. Okay, my dear?
Okay.
You took this last year, the different types of immunity. We'll revise it quickly. Active immunity, this is when your body produces antibodies. It's produced by the body. The antibodies are produced by the body. Either you're exposed to the infection itself, this is natural artificial immunity, or you take a vaccine. And we did talk about passive immunity, when you take ready-made antibodies, either naturally through breast milk or by taking monoclonal antibodies in the hospital. Is this diagram clear?
Yes.
Clear material. Okay. This graph is very important. You have time plotted on the x-axis and antibody concentration on the y-axis. And you will always be asked to describe this graph. This is first injection with a vaccine. So this will show you primary immune response. In the primary immune response, the graph is much shorter than the secondary immune response. Now, the reason is, I expect you to know it, when are the memory cells produced? In the primary immune response or in the secondary response? Where did we?
And the second response?
Yes. That's why the graph is much higher in the secondary response, because much more antibodies are produced and much quicker in a very short period of time. Because memory cells are already present in the blood, they are quickly activated, quickly they differentiate into plasma cells, quickly they produce antibodies, and the concentration of the antibody in the blood increases very, very quickly. That's why the graph is very steep in the second part of the graph. In the primary immune response, we still have inflammation and phagocytosis and antigen presentation, and then clonal selection, clonal expansion. This takes time. That's why it is much slow. And the amount of antibodies produced in the primary infection is much less. Clear?
Clear.
Good. You know what is a vaccine? The last thing we will talk about is the advantages and disadvantages. Also, this is last year material. Vaccines, they are expensive. They need educated medical staff. They can sometimes produce an allergic reaction. Vaccines sometimes can cause the actual disease if you have a poor or a weak immunity. Okay, this is the disadvantage of the vaccine. Expensive, sometimes it needs a booster dose, sometimes it may cause an allergic reaction. And by the way, this is very common, and this was very common in the COVID vaccine. A lot of people showed side effects. Sometimes it can kill the patient. Sometimes the patient will take the vaccine, and instead of becoming immune, he will become sick because his immune system is very weak. Okay?
Okay.
Passive immunity, this was also last year material. Passive immunity, here are memory cells produced. In passive immunity, when you take ready-made antibodies, hello? Hey. Hey?
Miss,
Yeah,
can you hear me?
now I can. Hello?
Miss, were you explaining or were you asking a
I was
question?
asking a question. What are the disadvantages of passive immunity? Does your body produce memory cells in passive immunity? Do you have lifelong immunity?
No.
No, this is the main disadvantage. Unlike any vaccine, it can cause an allergic reaction. And if you're immunocompromised, it can cause the type of disease. Now, I will leave this part because I will explain it in the lesson infection. I just want to go here to the type of vaccines because this is AS material. Either the vaccine that you take is a live pathogen, it's a living pathogen, but it is weak, or the vaccine that you take is a dead pathogen. It's a whole pathogen, but it is killed, it is dead, or part of a pathogen. Now, which one do you think is a stronger vaccine? The living pathogen, but has been weakened, or the dead pathogen, Aya?
The dead pathogen.
The dead pathogen is stronger or weaker?
It's weaker than the
Yes.
live.
Very good. And this is very logical. So the two types of vaccine, either you will take a living vaccine, a living pathogen, or a dead pathogen. Now, the living vaccine is very effective. It's very strong. But sometimes this can be a disadvantage because if you have a weak immune system and you took a living pathogen, you'll be sick. Okay?
Okay.
So this is the disadvantage of the living vaccine. You will take a whole pathogen that is alive. It's alive. You're putting a bacteria in your body, but it just has been weakened and that will multiply slowly. The advantage is that it is very strong. And if it works properly, it will give you lifelong immunity. The disadvantage is that it can make you sick. It may cause disease. Example, the MMR vaccine. Now, if you are an old person, if you know, I don't know if you knew this or not, when you went to take the COVID vaccine, there is one vaccine for people your age and one vaccine for people my age. Because my age, supposedly, I have a weaker immune system, so I cannot take the live vaccine. I took the inactive vaccine, the vaccine that is weaker, that is made up of the dead bacteria, the dead pathogen, or part of the bacteria, either the dead pathogen or its toxin. The advantage that it is safer. It will never cause a disease because it's actually a weak vaccine. But the disadvantage is that maybe it will not even produce any effect. It will not give you protection. It can be too weak. Okay, Aya?
Okay.
Good. Usually the inactive vaccine, because it's weak, it needs a booster dose. And both of them can give you an allergic reaction. Fine?
Fine.
Okay, I'm done with this lesson. It was an informative one and we took a lot of things. This lesson, number one, depends on understanding. If you understand it, it will be very easy to study. If you don't understand it, you will find it Chinese and you will not be able to study it at all. So is everything I said today clear?
Yes.
Are you sure? Is everything understood? Understanding is the first step in studying anything. Okay, is everything clear?
Yes.
Good. Again, I will tell you to study it. If you have any questions, ask me next time. Next time, I will finish infection. We finished the long lessons. Now the remaining lessons are the shorter ones. Let me see what lessons do we have. I think we have respiration. Respiration is very easy, very easy. It's exactly respiration last year with more pictures. That's it. Where are we? How many lessons do we have left? Because I want to finish in September. This would be very good. Your notes look like this?
Yes.
Okay. So how many lessons do we have left? I think 3. We have gas exchange, which is so easy, infectious disease, and we're done with this. We're done with this, we're done with this, we're done with this, and this, and this, and this, and this, and this. Oh, we have only 2 lessons. Very good. So we have only gas exchange and infectious disease, and then the practical will take around 2 or 3 sessions. Also, everything is easy. Okay, very good. Today is what, A? Today is Wednesday?
Thursday.
Okay, so Thursday, Friday, I'll meet you Sunday.
Okay.
Back we are. Okay, Sunday also at 11. I like this time, so we finish early. Study. We took a lot of things.
Okay.
Please, and solve, and solve, and solve. And always look at the mark scheme. Always solve in blue and correct in red. Because if there is a mistake and you write it in red, you will spot it just before the exam. When you come to revise, they will spot it just before the exam, and you know your mistakes, and no one does the same mistake twice. Okay, my dear? And if there is anything not clear, send me on WhatsApp and I will explain it. Inshallah, you'll take an A star. Okay?
Okay, Miss.
See you Sunday. Bye-bye.
Bye, Miss.